right, everybody. Just for the record, I am a minute late getting here today, but I'm not, whatever that says, 8 minutes late. And that's going to mess me up because I'll check that during the course of the class, and the class I'll think we're ending sooner than we should. So anyway, I'm going to have a rough morning, I can tell. Hey, no surprise, right? The next time you guys come here, Thursday, be prepared to take a test, right? Bring number 2 pencils. That's a really good thing to have in hand. We'll hand out the tests just like we did the last time. It's going to be those multiple choice questions. There are going to be 45 multiple choice questions. All you need to write down are the right answers to those 45 questions, your name, and some semblance, excuse me, some semblance of a student number, right? It doesn't actually have to be your number if you, whatever, don't want to. You want to, that Thursday can be your big chance to beat 007, all right? So whatever you want, but we need something that looks like a number. We need your name. Bubble in those parts, and we'll be in good shape. I'll also want you to look carefully at the form of the exam that you have. A1, A2, B1, B2, that sort of thing. Fill that out, and we should be in pretty good shape. The exams will get graded right after you turn them in. We'll probably get those posted by the end of the day on Thursday on Pilot, so you'll be able to see exactly how you did. Right after class is over, right after you're done taking the test, you can find an answer key on Pilot, and I encourage you to take your exams home with you and mark out what you had chosen as the right answer, so you can do that direct comparison yourself even before we post the scores on Pilot. I'm hopeful that we'll wrap things up a little bit early today in terms of the lecture outline that I want to go through, so that we can have a mini review session for maybe 10 or 15 minutes, and I'm hopeful that during that review session there will be a chance for you guys to ask me questions that you might have. If you have burning questions that I don't get to, I think your single best bet is send me an email. I'll respond. I'll have lots of opportunities to respond today and tomorrow and even on Thursday morning. You can try to catch me in my office. I've got a pretty tight schedule today filled with meetings and such, but if you catch me, I'll be happy to talk with you. I guarantee you I'll drop whatever I'm doing in an email. It's kind of rude, but I'll be in these I'll be in these meetings, and while other folks are talking, I'll be pecking away on my iPhone answering your emails, right? But you guys are number one for me. It'll be more important than the other things that I have to make a physical presence for. Any questions about the test, the lay of the land, that sort of thing? If you have specific questions, let's hold those to the end, but the format of the test shouldn't be a big surprise. Hey, you know what a really good idea would be? Look at the old tests, all right? That's going to come in really handy for you. There's a bunch of them available to you on Pilot. Check them out, all right? And I've told a few people, I think I've told a bunch of people, so I don't want this to be a secret. The way that I'm making up this test, here's how it happened. I was in San Francisco on Friday. You know, I left Thursday afternoon. I got there Thursday night, was involved with talks all day on Friday, and at 5 a.m. on Saturday, I flew back to Dayton. On my laptop, as I'm flying back, it's 7 hours of traveling to get back from San Francisco to Dayton. I copied the exam out of the back of the lecture notes and pasted it into a new document, a new Word file, right? And I looked at it and I thought, oh, this was a really nicely put together test. I like it. It's good. And I said, gee, but look, I've got like 6, 7 questions from students that have been submitted for extra credit. Can I plug some of these in? And I found a way to plug 2 student questions onto this test. Right? So good. 45 questions, 2 of them are new. I'm on a roll here, right? And I still had 6.5 hours to go, right, before I got back to Dayton. And so the time that I had in the span of 6.5 hours, you have to appreciate I took a little break for the, the snacks that they hand out as you're flying. And oh yeah, I'll have let me have some apple cranberry juice, or So there are a few pauses, but I was able to change in the ballpark of 35, 40 questions out of that 45 that you'll find in the back of the lecture notes. So there are a couple that you'll look at and you'll say, hey, what a lazy jerk. It's exactly the same as it was the last time that he gave a second exam for this class. That's my gift to you. If you look at last year's exam in the back of the lecture notes, a couple of those questions are going to look really strikingly familiar. Of the 35 or 40 that I've changed, I'd say half of those easily come from my rating other years' versions of the second exam for this class. All right? So again, come on, seriously. I'm at 31,000 feet. There's turbulence. What do you want me to do, right? So that's what you're looking at here. Look at those old tests. It's not just a matter of my being lazy, right? I really think there's some neat opportunities for you to learn some things from looking at those old tests. I try to set up the tests in such a way that when you look back at them, or when next year's class looks back at them, you'll actually learn something from looking at how the questions get put together and the kind of concepts that I'm asking you to focus on. So again, look at those old tests. But enough about that stuff. Let's finish off talking about some pretty cool stuff. It was an Austrian monk, Gregor Mendel, who got this ball rolling about genetics. I told you guys on Thursday of last week, he's the first to have given a really concrete answer to a question that has been perplexing mankind for probably as long as mankind had words to phrase a question. How is it that like begets like? Of course, everybody was interested in why it is that kids look and sometimes act like their parents, right? Mendel didn't go about trying to answer that question by studying human genetics. He instead used a much better model genetic organism, pea plants. And from the insights that he got from that very good genetic organism, he was able to answer those questions, the same questions, as they applied to humans, and tell us about human genetics, how things get passed from one generation to another. And so at the very heart of the study of genetics is this foundational set of studies that we call Mendelian genetics. Right? In a nutshell, let's go over what's up with Mendelian genetics, and then we'll get into some of the fancier stuff that gets tacked on to that basic concept of Mendelian genetics. And the fancier stuff are things like codominance and pleiotropy and epistasis. Again, we'll get to that before the end of today's lecture. So I showed you this slide on Thursday of last week. I emphasized to you how much better a model genetic organism pea plants are than humans or just about any other organism that you might think of. Quickly, they have short generation times. They don't cost much for upkeep. They have large numbers of progeny. There are few, I would suggest no, ethical constraints about what it is you can do in terms of forced mating experiments. Incestuous, do whatever you like. Nobody bats an eye when you're playing around with mating in pea plants. Right? So it's got this nice selection. Oh, and another nice thing is there were a number of characters that he could just go down to the farmer's supply store and purchase so that he could do his studies on. By characters, I'm talking about things like flower color, and seed shape, and seed color, right? Those are characters. The specific versions, white flower versus purple flower, those are traits. Okay, so he had a number of characters that he could study with just 2 traits for each of them. It was a perfect setup for him to be able to answer the questions that he hoped to be able to address. And compared to humans, again, just what a phenomenally good model genetic organism. People who came after Mendel learned the lesson about how to pick a good model genetic organism. And people have been able to do better than Mendel with his pea plants. Fruit flies, for instance, peg out on these scales relative to pea plants. fruit flies are an even better model genetic organism. Didn't occur to Mendel that that might be useful or helpful, but others subsequently found out that fruit flies have even shorter generation times, even larger numbers of progeny, have no ethical constraints for mating, little in the way of upkeep, and a number of characters that can be studied. So fruit flies are even better, but I'll tell you, pea plants were hands down better than anything that anybody had tried to study genetics with before Mendel, and that was the secret to his success. So he would do these mating experiments with pea plants. He started very simply asking, what's up with flower color? He took true breeding purple flowering pea plants and true breeding white flowering pea plants, and he mated them together. True breeding means that if you took those purple flowering pea plants and you let them breed amongst themselves, you would only get purple flowering pea plants. That's what true breeding means. And the same would apply for the white flowering pea plants. So the true breeding part was important, but he didn't do the true breeding thing. He took a purple flowering plant and a white flowering plant, and he forced them to have intercourse, as pea plants have such interaction. And he looked to see what kind of baby pea plants resulted. And what he found as a result of that mating experiment, in genetics you could call it a cross, what he found as a result of that mating experiment was a population of pea plants that he had the clever idea of calling the F1 generation, short for first filial. It's the first generation of baby pea plants that came out. He found an F1 generation from that mating experiment, from that cross, that was exclusively purple flowering pea plants. And it's the sort of thing that made him say, what happened to the white part? What's going on? And come on, you don't have to be a very deep thinker to say, seems like the purple is in some way dominant to the white. The white's getting, brushed aside, it's getting beat up, whatever. White's not showing up. Purple is the big kid on the block. He called it dominant. Pretty good choice of word, right? The purple trait is dominant relative to the white trait. But he wondered, what happened to the white trait? While it's being dominated by the purple trait, is it just gone? Does it leave town and you never see it again? Or is it masked in some way? Or is it blending in? Is it being coerced into becoming purple? What's up with the white thing? Is it just completely gone? And he had this neat next step that I think speaks to his curiosity and his intellect. It occurred to him, why don't I have these pea plants mate amongst themselves? It's kind of a neat leap that I don't know that I would have made. I would have, oh, gee, well, the white stuff's gone. So much for that experiment. Maybe I should move on to some other character. Let's play with seed color now, right? But Mendel had this neat idea. Let's play this out one step further and see what happens. So he took the F1 pea plants, all purple flowering, mated them amongst themselves, and this is what came out in the F2 generation, the second filial generation. The white flowers showed up again. How about that? Everybody roots for an underdog, right? The poor white thing was, recessive. It's getting beat up by the purple thing. His heart must have skipped a beat or two when he said, oh, look, white flowers, they're back. Hooray, good for you, you little white flower underdog. Come on, keep up the good work. But the white flowers, it wasn't an equal mix, right? It was 3 times as many purple flowers as white flowers. What's up with that, right? And the only way that Mendel could make sense of this as he was thinking about it is this, is there were 2 parents. What if each of the parents contributed a particle that dictated flower color? The true breeding purple guys must have contributed a purple particle, and the true breeding white guys must have contributed a white particle. And both of those particles came together and hung out together in another generation of pea plants that were purple flowering. The purple trumped whatever was going on from the white particle. But the two of them didn't mix, they didn't blend, it's not an average of each other. They stayed side by side in that next generation of pea plants unchanged. And then when that F1 generation started to make eggs and sperm, ovum and pollen in the context of a plant, but rough equivalents, when that next generation, the F1 generation, started to put together its gametes, one of two things happened. The gamete either got a purple particle or it got a white particle. And when those gametes came together, you got one homozygote, two heterozygotes, and one homozygote. Homozygote, homo, same. Homosexual, these are people who like to have sex with other people of the same sex, right? Homozygote, the zygote is the embryo. Homo, it's the same. It's got the same genes, or the same particles in Mendel's thinking. Heterozygote, heterosexuals, right? They like to have sex with people of the other sex, right? Hetero, different. The zygote has two different particles. So he had one homozygote purple flowering plant where it had both particles were the purple. He had two heterozygote purple flowering plants where there was one purple particle and one white particle. And he had a homozygous white flowering pea plant where both the particles must have been white flowering. Okay? Just like dear old grandma way back up here. Or grandpa. It's hard to tell sometimes with plants, right? So that's how he rationalized the whole thing. It's the only way this made sense. It was important. There were two really neat insights that he got that cracked open all of genetics for all the rest of diploidy, right? All the rest of the organisms that are diploid, like you and I. One, there are particles that are dictating these traits for each of the characters that he was going to study. There were particles. We call those particles genes. He didn't have, he was 100 years before, 75 years before people started talking about DNA being the genetic material. So there was never an opportunity for him to use the word gene. He was talking about particles, but he meant genes. In our translation, we would translate his particles to genes. So the first neat insight he had is particles. And the second is that there were two particles in each organism. All organisms get two particles, one from their mother, one from their father. And when it's time for them to have babies, they pick randomly between those two particles. They either pass on mom's particle or they pass on dad's particle. Not both, and not neither. One of the two. So his two neat insights are particulate inheritance and this idea of diploidy and then segregation associated with it. So that's how these numbers ended up making sense to him. Conceptually, that's what he's got in mind. And he puts it to the test with what? I think we counted these 9 different characters over the course of the next 10 or 15 years that he did his experiments. All right, so he started looking at flower color, purple flowering and white flowering. He said, ooh, that's cool. I wonder if I'll find the same thing happens if I look at flower position, axial versus terminal. Right? How about that? I get the same thing. I get the same 3-to-1 ratio, more or less. What about seed color? What about seed shape? Everything that he looked at, he got essentially a 3-to-1 ratio for the traits that were displayed in the F2 generation. He always started out with true breeding plants with 2 different traits. He always found that in the F1 generation, he'd get just 1 trait. He'd call that the dominant trait. And he always found in the F2 generation, 9 times out of 9, he found in the F2 generation that he saw both traits appearing and that they would be in a 3-to-1 ratio. Dominant 3, recessive 1. All right? So he sees this time and time again, and he figures he's got this worked out. So that, in essence, is Mendelian genetics. And it really comes down to this idea that there's particulate inheritance and that we're talking here about two particles that come together and that they get segregated independently of each other. So with that sort of conceptual framework, others come along and flesh out the theory. But I'll tell you, the next couple steps are trivial compared to the flashes of insight that Mendel had to get this far. To talk the rest of the way through this lecture, I need to, before I tell you about these next add-ons to Mendelian genetics, let's just get some vocabulary out of the way. I want to make sure that we're all using the same terms and there's jargon that's involved here. You guys know about chromosomes. Those are big chunks of DNA. Let's wrestle with this idea of a homologous chromosome. Homo, same, right? Logous, who knows what that comes from. But homologous means it's the same, it's the equivalent chromosome. We each have 46 chromosomes in all of our cells. 22 autosomes and a sex chromosome present, 2 copies each. It'd probably be easier if I just told you this. We've got 2 copies of human chromosome number 1 in all of our cells. You know what we call those chromosomes, the copies of chromosome 1? We say we have a pair of homologous chromosomes. We have a pair of homologous chromosomes number 1. So that's what a homologous chromosome is. It's my mom's copy of chromosome 1 compared to my dad's copy of chromosome 1. So we have homologous pairs of chromosomes. And on both my mom and my dad's copy of chromosome 1, we'll have the same arrangement of genes. Now, I don't expect that I'll find I have a gene for flower color on my chromosome 1, but if we bring this now back to Mendel, right, on a chromosome in pea plants, there's a gene that's responsible for flower color. On a homologous chromosome, you may, on homologous chromosome pairs, you may find that you have 2 different versions of the gene. Okay, you may have a version that codes for purple flowers, or you might have a gene that codes for white flowers. Those different variations of the gene are called alleles. In England, you would call it an allele, right? But gosh darn, we're in America, they're alleles. Okay, and there are more of us than them. We're going to win in this battle over the course of time. So if you can detect a difference between 2 versions of a gene, that means that there's an allele. Okay? If the 2 versions of the gene are exactly the same, you know what you got? You got 2 copies of the same gene. If you have 2 versions that are a little bit different, if you can recognize the difference between them, now we start talking about a pair of alleles. There's no limit to the number of alleles you might find for a particular gene. You might find there's only 1 version. You might find that there's 2. You might find that there are 550 or some other number, right? But if there's a distinguishable difference between one version and another, we say that we have at least 2 alleles. And the location on the chromosome that the gene resides is called the locus where it resides. Okay? Locus is the Latin word for location. In fact, the word location has its root, and it sounds like locus, because that's how we got the word location in the English language. Okay, so locus means location. It's just a location on the chromosome. Did you think you would get a Latin lesson in a biology class? Let me teach you one little bit of Latin. The plural of locus in Latin is loci. Again, that's how Americans pronounce it. The correct Latin pronunciation is loci, all right? The British say loci, we say loci. We're wrong, they're right. There are more of us, we're going to win. All right? So when you're talking about more than one location, like the location of 2 genes, right, we start talking about loci. When you're talking about a single location, we talk about a locus. Okay? And at a locus, you may find that you've got an allele or 2 or 3. All right, so there's the jargon associated with connecting this now from Mendel's way of thinking about particles to our modern understanding of how it is this all plays out. We know that what's really going on is at the heart of the particles that Mendel got so excited about is DNA. DNA hangs out in big chunks called chromosomes. Along the chromosomes, we find a number of genes. Those genes can have alleles. They hang out at specific locations. They reside at a specific location along the length of a chromosome. That location is a locus. All right? So the modern day jargon associated with Mendelian genetics. And again, I told you that after Mendel has this neat breakthrough where he's figuring out about particulate inheritance and the segregation of these alleles, others come along and they put some math to it and they dress it up and they help come up with ways to make it easy to predict what sort of combinations of traits you might find. Mendel himself had kind of figured this slide out, right? And I had already telegraphed this to you. Let's go back a couple of slides. Remember when back here I said there was a homozygous purple flowering plant, two heterozygous purple flowering plants, and a homozygous white flowering plant? Keep that mental picture. Here it is, again, just arranged a bit of a different way. Remember that 3-to-1 ratio. In fact, that 3-to-1 ratio is often referred to as a classic Mendelian genetics ratio. 3-to-1. When somebody says 3-to-1 to somebody who's involved with biology, first thing that pops to mind is, classic Mendelian genetics. There's a dominant allele and a recessive allele, and we're talking about effectively an F2 generation. Okay? 3-to-1, just lights start flashing. That sounds an awful lot like a single gene that has two alleles, one's dominant, one's recessive. So 3-to-1 is a classic Mendelian genetics ratio. I'll also tell you another ratio you see a lot in genetics contexts is 1 to 2 to 1. The ratio of homozygous, dominant purple flowering, to heterozygous purple flowering, to homozygous white flowering pea plants. So the genotypic ratio, the types of genes these organisms have, has a 1 to 2 to 1 ratio. The phenotypic ratio, the way that these plants look on the surface without necessarily knowing what their genotype is 3-to-1. 3 purples to 1 white. Okay? And then, as I said, others come along, they say, ooh, this Mendel guy's onto something, and they dress it up a little bit. There's this guy named, I think his first name was Arnold, it might be Arthur Punnett, comes along, and we today call these things Punnett squares because of this guy's idea of how to represent what's going on with Mendel's particulate inheritance. The way he sees it is this. It's all coming down to probability. Pretty simple stuff. If I were to tell you what, this is one of the few times that the money I carry with me comes in handy. I have a silver dollar, right? What do you think? What are the chances that if I toss this silver dollar, I'm going to get heads twice in a row? I'm hearing a lot of people coming up with a good answer, right? About 1 in 4 chance that if I toss it twice, it'll come up heads both times. You know how you figure that out? I don't think it's from looking at the Punnett square. I think you figured out, gee, you got a 50% chance of getting heads the first time, a 50% chance of getting heads the second time. The chance of both of those being the same is 1 in 2 times 1 in 2, or 1 in 4, right? What we've just done now, what you guys did intuitively, and what I've just talked you through, that's called the product rule. You're multiplying together these two probabilities. You're taking the product of the two probabilities, and that tells you the chance that both will happen together. So, you know, what's the point of holding the silver dollar if you're not going to toss it a couple times? Tails. So I'm a loser right out of the gate. We don't need to continue that experiment anymore. But, so here's the deal. If you think about this segregation of the particles as a coin toss, check out what happens, right? There are going to be some organisms that spend their time making sperm, all right? And there are going to be other organisms that spend their time making eggs. Now, if these organisms happen to be in the same species, the egg and the sperm can get together to make a zygote, a little baby organism that can grow up to be in a big organism that spends its time making eggs or spends its time making sperm. Right? But for the organism, if it's a heterozygote and if it's making sperm, what are the odds that a particular sperm that you choose has, who even knows what capital R stands for here, but what are the chances that it gets a capital R as opposed to a lowercase r? It's a 50-50 chance. Any sperm you choose, any sperm that gets made, half of them are going to have capital R, half of them will have lowercase r. And if the female is heterozygous as well, when she's making her eggs, half of her eggs are going to have the capital R, and half of them will have the lowercase r. And the way you're supposed to think about these Punnett squares is this. What if one of these sperms with a capital R bumps into one of these eggs with a capital R? We get a diploid zygote that has two capital R's, right? In contrast, if one of these sperms with a lowercase r bumps into one of these eggs with an uppercase R, you get a heterozygote. Mom's uppercase R, dad's lowercase r. But you could always have the situation where one of dad's uppercase R sperms hooks up with one of mom's lowercase r eggs, get a different kind of heterozygote, dad's capital R, mom's lowercase r, or you might once in a while get mom's lowercase r egg to hook up with dad's lowercase r sperm, and you get a homozygous lowercase r, right? So that's how this Punnett square works. And that's how it all plays out. If you guys look at the back of the lecture notes, you'll see a copy of Exam 2 where the last 5 or so questions are asking you to think your way through some mating experiments. Okay? A Punnett square written on the margin may well help you quickly and easily figure out what the right answer is. One of the questions I remember from last year's test was something about dwarfism, right? Turns out that achondroplasia, one of the ways that one can become a dwarf, is actually a recessive lethal trait, right? So if you have 2 copies of the achondroplasia gene, that translates into a medical condition that we are all familiar with. It's called dead, okay? The only way that the people who have achondroplasia, dwarf, small stature, right? They're not homozygous for this recessive gene because the recessive gene is a recessive lethal. If you got 2 copies, you're dead. We don't see those people. They don't get born, right? They're not around for us to be seeing. The only people that we see that have achondroplasia are people who are heterozygous. And people who don't get from either parent that recessive lethal gene are homozygous. They don't have achondroplasia. So what would the ratio be of kids to a couple, a pair of people who have achondroplasia? What would be the phenotypic ratio that you would expect? That's a question straight out of the 2013 test for this class. I'm telling you, achondroplasia is a recessive lethal. And what that means is for homozygotes, we could put a big red X there. We're not going to see those folks because they die before they get born. But the people, the mom and the dad's got 1 copy of the gene that is not going to make the disease state or the achondroplasia, and he's got 1 copy that is going to cause that particular set of phenotypic characteristics. And the mom's going to be the same way. I'm telling you, this fertilized egg, this zygote, is not viable. So we don't see anything here when we're talking about adult individuals or children. But this person and this person will have achondroplasia. So there's going to be two of them. If you're a heterozygote, if you have one lowercase r and one uppercase r, you are going to have achondroplasia. And if you've got two uppercase r's, you're not going to have achondroplasia. You're going to be a normally sized individual. So what's the ratio?
Dr. Crane, it has to do with dominant, not recessive, or does it play out with this?
You know, here, we're going to get into dominant and recessive a little bit more later on. It turns out that in this circumstance, we would refer to it as a recessive lethal, right? You might, it's not even strictly dominant or recessive, it's kind of codominant, all right? But for this purpose, let's talk about how the ratios would play out. Well, let's say, what are the odds that this couple will have a normal-sized child? It's about 1 in 3, because we won't see one of those children. For every normal-sized kid they have, we expect they're going to have 2 dwarfs. Okay? What are the odds that they'll have somebody with achondroplasia? 2 to 1, right? 66% chance. All right, so again, what I'm hoping you guys can do is when you're confronted with that kind of question, and I can give it to you in a couple of different ways. I can tell you a family or a litter of cats has, you know, this color coat for their kittens and such. Think about how it all fits into one of these Punnett squares, and I think you'll find that you can answer just about any basic Mendelian genetics question. Okay? So these are Punnett squares, and this is the principle. Bottom line is on the top and on the side, you're drawing out what sort of gametes the organism can make. If you're a homozygote, that means that both gametes are going to be the same. If you're a heterozygote, that means that one gamete will be one, and the other gamete will be the other kind of allele. And then you're just mixing them together. This cell is the combination of this one and this one. This cell is the combination of this one and this one. In pea plant parlance, here's what's going on. Now with a Punnett square there to round out Mendel's perspective on things. Mendel didn't have the advantage of a Punnett square, but this is how it would have played out if he had one to play with. What he was doing, he was taking true breeding purple flowering plants. True breeding is synonymous with homozygous. Okay? Homozygous means 2 copies of the same gene. Okay? True breeding purple flowering plants, homozygous dominant. 2 true breeding white flowering plants, homozygous recessive. Come together, when you mix those gametes together, you're going to get heterozygous F1 plants. The dominant trait will, for lack of a better word, dominate and manifest itself. So these will be purple flowering plants. The recessive trait will not manifest itself, but it's maintained throughout the life of that pea plant, unaltered in any way from its close association with the purple flowering gene. When you mate these F1 plants together, the important thing to bear in mind is you're mating heterozygotes, right? The heterozygote has 1 dominant allele, 1 recessive allele. When you're making sperm, some of those will have the capital P, some will have the lowercase p. When you're making eggs, some of those will have the capital P, some will have the lowercase p. And when you plug that into something like a Punnett square, check out what you get. You get a 3-to-1 ratio, 1, 2, 3, of purples to whites, and you have a genotypic ratio of 1 homozygote purple to 2 heterozygous purple to 1 homozygous white. Okay? So wrapping it all up, that's what Mendel played with. And again, there's a couple of really simple concepts here. Let me underscore them. Particles are what's going on, right? We'll call them genes. We can talk about different versions of the genes called alleles, but boy, wrap your head around this. It's particulate inheritance. It's individual genes that are dictating these things. And another important concept is this idea of independent assortment or segregation, right? Having a purple allele in the same cell as a white allele doesn't change the purple allele or the white allele in any way. When time comes to make up gametes, those are passed on as if they had never had a brush with the other allele. The alleles don't change, and they assort independent of each other. If you get those two concepts, all the rest of this stuff follows, and it arms you with an ability to do some pretty slick stuff. Now, I suspect this has not been a burning question for any of you, but it came up for Mendel a couple times. He found himself once in a while, and you can imagine how this might apply in your own lives. It's not that big of a stretch. He found himself once in a while wondering what particles did his peas have. He lost track. They got mixed together. He could test genetically to figure out what particles were in the pea plants. You never need to test for a homozygote recessive, right? Because why do you want to test what the genotype of a homozygote recessive is? You know what it is. Look at it. It's showing you the recessive trait. The only way you can see the recessive trait is if it's a homozygote. Homozygote means both particles are the same. So for Mendel's white flowering plants, there was never a mystery what the genotype was. If you had the white flowering phenotype, you knew immediately what the genotype must be. 2 lowercase p's in this case. But if he had a purple flowering plant, that's the sort of thing that could happen in one of 2 different ways. Now, odds are, if this is the result of an if he's got F1 pea plants, the odds are twice as good that any purple flowering plant he picks is a heterozygote than a homozygote, but you can't be sure, right, which it is. So if you give me an F1 progeny from one of his pea plant experiments for flower color, if you give me a bunch of F1 pea plants and they're purple flowering, I don't know. I think if I had to bet, I'd say it's a heterozygote because the odds are in my favor, but I certainly am willing to entertain the possibility that it's a homozygote for the dominant trait. So how do you tell? How do you tell who should get the payoff on that bet if you're taking that bet? Well, you do a test cross. And what you can do is you can take one of your white flowering plants for which you know what the genotype is and mate it with the suspicious purple flowering plant. And one of 2 things is going to happen with the baby pea plants that result. Sometimes you'll find you get nothing but purple flowering F2 pea plants. And sometimes you'll find that half of your F2 pea plants are purple flowering and half are white flowering. The difference is simply this. If this purple flowering plant is a homozygote for the purple flowering gene, all of the progeny of that cross are going to be heterozygous. Heterozygotes will manifest the dominant trait. They're going to be purple. However, if this purple flowering plant is a heterozygote, meaning it's got 1 capital P and 1 lowercase p, half of the progeny of this cross will be purple flowering heterozygotes and half will be white flowering homozygotes. So it's a test cross. You can use this sort of experiment to determine what the genotype of an organism is when all you've got to work with is the phenotype. All right, so we've already touched a little bit on this idea of dominant and recessive and how sometimes the waters can get muddied. A truth in advertising sort of thing for you here. I expect most of you have been exposed to Mendelian genetics before I started talking with you about it today. I know you got some exposure in the lab last week, but probably in high school, maybe even before high school. But I wonder if anybody has bothered to tell you some of the dirty secrets associated with Mendel. He's a monk, right? So they can't be too scandalous. But Did you know that there's been, in the last 20 or so years, people who have been suggesting that Mendel may not have been as honorable as you might have thought a monk would be? The only results that Mendel ever talked about publicly were the ones that fit really neatly into this Mendelian genetics sort of scenario. The 1 to 2 to 1 genotypic ratio is a 3 to 1 phenotypic ratio. Whenever he experimented with something that didn't quite fit that mold, he just sort of brushed it under the rug and never talked with anybody about it. For instance, he would sometimes try to keep track of 2 traits at the same time. Okay? Not just flower color, but flower color and seed shape, for instance. That gives rise, those crosses, in Mendel's thinking, should give rise to a ratio of 9 to 3 to 3 to 1 for the F2 generation. I'll show you a picture of that Punnett square in just a minute. Right? But that only happens if those genes are on separate chromosomes. If they happen to be tethered together on the same piece of DNA, you don't get 9 to 3 to 3 to 1, you get something else. Whenever Mendel got that something else, okay, let's not talk about that one. Right? And again, Mendel found himself occasionally going down to the farmer supply store and buying pea plants that didn't obey this simple dominant and recessive sort of relationship that we've been talking about. There's something else that was going on apparently with the inheritance. It could be codominance, it could be epistasis, it could be pleiotropy. He never talked about those. Right? And so he's gotten some heat in the last couple decades as people have looked back at his notes and what are the actual experiments that he did. In his defense, that was standard practice of the day for people doing scientific research, right? They thought of themselves as advocates. They had to build a case and defend it against others. So that was their job to give the evidence in support of their case, and it was left to others to come along later on and show outliers or exceptions to the rules, right? But so again, Mendel occasionally encountered some things in his genetics experiments that just didn't quite make sense. He never talked about them publicly, but we can tell from looking at his notes that there were occasions where things didn't fit neatly into this simple dominant and recessive sort of thing like we've been talking about with flower colors. For instance, there's this thing called codominance. Okay? In snapdragons, flower color is determined by a single gene. There's a bunch of genes, but for this particular pair of flower colors, there are 2 alleles. At this locus, for this gene, there are 2 alleles. One allele causes the snapdragons to have red flowers. An alternative allele causes the flowers to be white colored. Okay? What's dominant? What's recessive? Well, here's the thing. If you have a true breeding red flowering snapdragon and you made it with a true breeding white flowering snapdragon, the F1 generation's flower color is neither red nor white. It's a hybrid. It's an intermediate. It's pink. Right? So that kind of relationship is fairly common for alleles. We see that with the achondroplasia example that I mentioned before, right? If you have 2 capital R's, let's make it capital A's for achondroplasia, that makes you a normal-sized individual. If you've got 2 lowercase a's for the achondroplasia gene, that makes you dead, right? And if you've got a capital A and a lowercase a, you've got this intermediate trait in some sense. Both of those genes are manifesting themselves in what the phenotype of the organism is. You got that dwarfish sort of appearance. What's happening here with these snapdragons is, again, an instance of codominance. Both of those genes are equally strong in terms of showing up in a heterozygous individual. So for the P generation, we have true breeding red and true breeding white snapdragons. For the F1 generation, you're going to get heterozygotes, nothing but heterozygotes, and they will have a phenotype that is intermediate or pink. And for an F2 generation, now the phenotype, the way that the organisms look, again corresponds directly to the genotype. Pink flowering snapdragons in this scenario are heterozygous, red flowering are homozygous, white flowering are homozygous. Okay? So this is how codominance plays out. Both of those traits are being manifest in the heterozygotes. Only the red trait is manifest in homozygote reds, and only the white in homozygote whites. Here's a more human example of codominance. I'll bet everybody in this room knows what their blood type is. I'm AB negative, and I'm proud, right? It's the rarest of all blood types, I'll let you know, right? And it's shared with a number of prominent individuals like Abraham Lincoln and George Washington. So fine company to be with. Something like 0.5% of the North American population is AB negative. So I've got the best blood type of anybody, but I suspect you probably know your own blood type. Blood type, the ABO part of blood type, is a nice, neat human example of codominance. There's a single gene that determines that AB part of our blood type, okay? That single gene has 3 different alleles. There is an allele that gives rise to an antigen on the surface of our blood cells called, get this, the A antigen. And there's another allele that gives rise to a different antigen on the surface of our blood cells. That's the B antigen. And there's a third allele that just says, I messed up. I'm not going to give rise to any antigens. It's the O, kind of a big 0 allele. So there are 3 alleles. There's the allele that says, don't put anything at all on the surface of the blood cell. There's something that says, put an A antigen on the surface, and there's something that says, put a B antigen on. Again, I'm AB negative. The negative is for a different time. I'm a very positive guy, but my rhesus factor is negative. I don't display the rhesus factor, but that's a separate story. I'm AB. I'm telling you not only what my phenotype is, that's also my genotype. I must have gotten from mom an A antigen allele, or maybe the B antigen allele, and whatever I got from mom, I got the other from dad. I got an A antigen, I got a B antigen, I got one allele that says make A, I got one allele that says make B. Codominance. For those of you who are blood type O, I know what your genotype is. Your genotype is homozygote O, right? For those of you who are type A, I'm kind of scratching my head about you guys, because you could be homozygous for A, 2 alleles for the A, or you could be a heterozygote where one of your parents gave you the O and the other parent gave you the A. And the same applies for those of you who are blood type B. Right? But if you're O, I know you're a homozygote. If you're AB, I know you're a heterozygote. I know exactly which heterozygote you are. Right? And if you're A or you're B, I got to do a test cross. Anybody want to sign up for that experiment? It's not, I probably can't get around to doing it. But so blood types in humans is a neat example of codominance. Fair enough? So where have we been? We've talked about the nice, simple, clean-cut Mendelian world of dominant and recessive. And now we've begun to explore this stuff can get a little bit more complicated from things like codominance, right? Sometimes both of them show up at the same time. We've seen that in the snapdragon flower color. We've seen that in human inheritance for blood types. But Well, and the human blood type also gives you a sense of this, too. There's sometimes more than just one or two alleles. For our blood type antigens, there's 3 alleles for that particular gene. When I say multiple alleles, I mean there could be a whole bunch. I've already mentioned the number 550. Pick whatever number you like. You could have a gene with an enormous number of different alleles. It also turns out that characters in complicated organisms like you and I, characters, features like hair color, eye color, skin color, athletic ability, dribbling skills, IQ, alcohol tolerance, those types of characters in humans are almost always governed by more than one gene. Right? So not only can you have genes that have multiple alleles, we can have characters, when we're talking about us, that are determined by multiple genes. In pea plants, flower color, it's a one-trick pony. There's one gene that's responsible for purple versus white. In humans, all of those traits that I just rattled off, some of them might have gotten your attention. Skin color, IQ, some of those, well, all of those traits are determined by multiple genes. Let's make this less personal and talk about coat color in rats. Turns out that there are 2 genes, each with 2 alleles, that govern something like 90% of what determines the fur color for a rat. Okay? It's most of the story. And what ends up happening is this, is if you do a mating experiment between these 2 heterozygous individuals, notice there's 2 genes. There's the B gene and the C gene. This guy, I'm a biology professor, I'm looking for testicles here. I don't see any. So I'm thinking this may be the female. This female crossed with this male who's being very polite and hiding his testicles. When you mate these 2 homozygotes, when you mate these 2 heterozygotes, look what happens. You can get some progeny that are capital B, capital B, capital C, capital C. Whoa, that guy's dark, right? But you might end up with some lowercase b and lowercase c hooking up with a lowercase b, lowercase c, this sperm and this egg, and whoa, that guy's not so dark. That one I think we should just call white and be done with it, right? So look at the range that you get there. This is now a dihybrid cross. We're looking at 2 traits at the same time, but it's not really a dihybrid cross because we're looking at the same character. And if you have just a 2-gene system with just 2 alleles, look at the different range of coat colors that you can get. You can get full-blown black, you can get this sort of intermediate thing, and you can get white. And don't forget, well, and so there's this Mendelian ratio that comes into play here as well. So these are our simple dominant recessive relationship that's going on, but there's 2 genes that are in play at the same time, and they both have an equal say. My point is pretty much simply this. Many traits, many characters, I should say, many characters are determined by more than one gene. A character that's determined by 2 genes is a lot harder to make sense of than a character that's determined by 1 gene, at least from a Mendelian genetics perspective. A character that's determined by 3 genes, this is getting nasty pretty fast. It's going to be really hard to tell what the genotype of an individual is from the phenotype if there are 3 genes involved, even if there are only 2 alleles for those 3 genes. Things like IQ are probably determined by on the order of a dozen genes. It gets really hard, and each with many alleles, it gets really hard to make solid predictions or even put odds on how smart the kids of a particular pair of humans will be because of this polygenic inheritance. There are a number of genes that are going to be influencing that same single character. So here's an example of polygenic inheritance. Here's what happens when you get 3 genes that are involved. Again, there's just 2 alleles for these 3 genes. Many times you'll find multiple alleles that just muddies the waters still. But if you've got a trait or a character that's determined by 3 genes, look at the range of phenotypes that you end up with. And now they're trying to move this more into the realm of human skin color. That can account for why it is that 2 medium dark-skinned people might have kids that are light-skinned and dark-skinned. That sort of thing can happen. And when you see that sort of thing happening within your own families for different traits, what you probably need to start thinking about is that you don't need to think about adoption or, switched at birth kind of things. You need to start thinking about, oh, I'm looking at a character that's probably determined by more than 2 genes. Maybe 3, maybe many more. All right, so let's talk about the different kinds of things that we've seen so far. Dominance of one allele over another. Mendel played with that stuff a lot. Incomplete dominance, or if you prefer, also incomplete dominance is when both traits show up equally. Codominance is the phenotypes. The difference between incomplete dominance and codominance, to me, isn't worth wrestling with, so I'll use those terms interchangeably. Appreciate, though, that there are other circumstances where you can have multiple alleles, not just 2. You can also have multiple genes. That complicates things quite a bit. And let me say a few words to you now about the next one on the list, this idea of pleiotropy. It's a nice sounding word, right? Pleiotropy. Pleiotropy is the word that we use to describe a circumstance when a single gene is actually influencing a whole bunch of different characters. So there's a gene, the example they're giving you here, that determines if one will get sickle cell anemia or not. To get sickle cell anemia, well, there are 2 alleles that are involved here. There is the normal hemoglobin gene, and there is the sickle cell version of the hemoglobin gene. If you have 2 of the normal, you don't have sickle cell anemia. If you have 2 of the sickle cell anemia genes, you have a full-blown, hardcore case of sickle cell anemia. And if you're a heterozygote, you have some sort of intermediate symptoms of sickle cell anemia. But the point for this pleiotropy is this, is that one gene, a single nucleotide difference at the level of DNA, that one gene ends up giving rise to a whole suite of manifestations in people with sickle cell anemia, right? They have difficulty clotting, they have inflamed spleens, they have thromboses, they have varicose veins, right? There's a whole bunch of things that are governed by that one particular gene or influenced by that one particular gene. So a pleiotropic gene is a gene that shows up when it's present. It changes a whole bunch of things. It doesn't affect just flower color. It affects flower color, seed shape, seed size, seed color, a whole suite of characteristics. So that's what pleiotropy is. And let me tell you about this idea of epistasis. Epistasis is the word that we use to describe the circumstance where a gene actually controls the expression of other genes. Right? So it's not so much that the gene is responsible for the character, but the gene regulates the expression of genes that are responsible for a character. So pleiotropy is when the single gene changes a whole bunch of characteristics of an organism. Epistasis is when a single gene influences the expression of a bunch of other genes that in turn each change some characters. So you can see how this stuff gets complicated pretty fast. And I've already tried to talk with you about this idea of polygenic inheritance. That messes things up in a big way in terms of trying to make sense of the heritability. Let me finish this lecture outline with just a couple words about this burning, eternal question of how important is nature versus nurture. The nature versus nurture argument boils down to this. How much of what we are let's make this personal, what you and I are, how much of what we are is governed by our genes, that's the nature, as opposed to how much is governed by our environment. Okay? I'll give you a specific example. We know that there are some genes that predispose people to alcoholism. Okay? We know that there are some genes that are involved with that particular trait. In this part of the world, if you have the right combination of genes, you may well be at very serious risk of being an alcoholic, just because of that combination of genes in the environment in which you live. That same person who in this environment is at risk of alcoholism, if they were living in Saudi Arabia, where alcohol is prohibited, right, that environment makes them much less at risk of becoming an alcoholic, because they just won't encounter the alcohol there. So their environment is different. That's a nice, clean example of nature versus nurture. But boy, you know, I wrestle with this. I'm sure you guys wrestle with this. How much of your success can you claim credit for, and how much do you need to thank your parents for? The fact that you're a certain height, the fact that you have that really nice hair color that, you know, we all appreciate, and thank you for sharing with us every day. You know, how much of that nice hair color that you have is because your mom and dad got just the right combination of genes that mixed together to give you that, as opposed to the fact that you went to the beauty salon and, you know, had the environment, you know, influence your hair color, or that you just were out on a nice sunny day and that bleached your hair a little bit and gave you those nice highlights. I mean, how much of that, you know, for things like hair color, yeah, we often have a pretty good guess. But what about stuff like intelligence, right? You guys are learning stuff. You're in an environment where you're learning things, right? You can't, that's not all your parents' good genes. There was an environment thing there too. So nature versus nurture is a very difficult question to tease apart. One of the best ways that we can determine how much of what we are is determined by our genes as opposed to our environment is, in humans, is by twin studies, by looking at individuals, identical twins that have been separated at birth, all right? And there are remarkable things that we've discovered about human behavior and human appearance through these types of twin studies. During adoption processes, sometimes the twins will just, by chance, be separated. Behavior turns out to have a strong genetic component, and aspects of behavior that you might not have thought would be determined by genes are heavily influenced by genes. I'm going to give you one example that'll blow you away, and then we'll wrap this up. Underwear preference in twin studies turns out to be heavily dictated by one's genes. And there apparently are 3 different kinds of underwear preference. I'll let you guys figure out what they may be. But if you find one twin in a twin study, I'll give you one of them. Likes to go, when I was growing up, it was called commando, right? If you have a twin that you find likes to go commando, you can predict with 90% confidence that the twin who's never met this other twin also likes to go commando. How about that? As opposed to either of the other 2 choices that one might have with respect to underwear preference. And it's not, the other 2 are not boxers and briefs, by the way. All right. So in lab, you guys played these Mendelian genetics games with pedigrees, more or less. Let me just tell you a couple things quickly about pedigrees. In pedigree analysis, a box is always meant to portray a male because they have sharp angles and poking things out. Females are pleasantly round, right? And so females in pedigrees are always shown as round. If you manifest a trait, the symbol is filled in. If you don't have the trait, it's left blank. And from looking at these pedigrees, you can often make some pretty good predictions about what the genotype of individuals will be just based on their phenotypes. There's a chondroplasia for you right there. Okay. That was a nature versus nurture, by the way. Same genes, different environment for hydrangea flower color. Don't forget, there's a test here on Thursday. I think we've got about 10 minutes left. So, the clock is right now? It wasn't right before. Well, my battery ran out too. I guess I'm out of time. How much time do you guys think we've got? We got 10 minutes. Well, it was rude of you guys to cut off my mic. I'll tell you this, I've got a corded mic, but there's nowhere for me to plug it in, so I just have to shout. The Tegrity part is still going to be working, so I'm shouting now for Tegrity, but I've run out of batteries, apparently, for my cordless mic. So let me shout and tell you what it is that I think are big deals about each of these lecture outlines that we've talked about since the last test. All right, I started talking with you about chemical energy in cells. A big part of that are the first and second laws of thermodynamics. Energy can't be created or destroyed. Law number 1, entropy increases. Law number 2. Spontaneous reactions are ones that go from a high Gibbs free energy to a low Gibbs free energy. Okay? So delta G is negative for a spontaneous reaction. The only way you get a reaction to go, or products to go from low delta G to high delta G, is if you couple that reaction to a more energetically favorable reaction in the other direction. Enzymes do their thing by reducing the activation energy of the chemical reactions that they catalyze. All catalysts work by reducing activation energy. That's it. That's the magic with catalysis, reduce activation energy. And activation energy is the amount of energy you have to invest to get the substrates to make a transition through a transition state. Okay? There's a picture that talks about activation energies. Say no more. Then, with energy in mind, we started talking about metabolism. Metabolism is the sum total of all the chemical reactions that go on within an organism. We can parse it into two halves. There's catabolism and anabolism. Catabolism is breaking molecules down from complicated large things to simpler small things. Anabolism is the flip side of that. Anabolism is when you're building stuff up. I started talking about catabolism, and a big part of that is glycolysis. Glycolysis is that 10-step process by which you go from glucose to pyruvate. Glucose is a 6-carbon molecule, pyruvate is a 3-carbon molecule. Along the way in glycolysis, you get a net gain of 2 ATPs. Energy can then be taken out of the pyruvate, largely through the citric acid cycle. Glycolysis happens in the cytoplasm. The citric acid cycle happens inside of eukaryotic cells, at least, in mitochondria. The citric acid cycle is a cycle. It goes around in a circle. Pyruvate comes in as acetyl-CoA. A CO2 gets lost as part of that. And then the acetyl-CoA, a 2-carbon molecule, ultimately gets converted into carbon dioxide gas, 2 carbon dioxide gases. And as you crank through the cycle, you make some ATPs, you make some NADH, and you make some FADH. The energy gets harvested from those NADHs and FADHs through an electron transport chain in the mitochondria. And that ultimately sets up a proton gradient that can be used to drive ATP synthesis in a process called chemiosmosis by an enzyme called ATP synthase. So again, this is the Reader's Digest version. 30 seconds or less for a hour-and-a-half lecture. Here is a picture that shows sort of all of that stuff at one time. Again, glycolysis in the cytoplasm, citric acid cycle, and chemiosmosis in the mitochondria. All of it is about capturing ATPs from the breakdown of glucose. That glucose gets generated, for all intents and purposes on this planet, by photosynthesis. Chemiosmosis can take place in deep sea vents, but you and I are relying upon energy that's coming from the sun. Ultimately, that's being captured during the photosynthetic process. Photosynthesis can get broken up into two components. There are the light reactions and the dark reactions. The light reactions are the ones where chlorophyll is absorbing photons and capturing that photoelectric energy and converting it into a chemical energy, synthesizing things like ATP, NADPH, and such. The dark reactions of photosynthesis take those energy-rich compounds and use them to synthesize glucose from cranks of the wheel of the Calvin cycle. So the dark reactions are the Calvin cycle, where you're doing carbon fixation. The light reactions are where you're capturing that electromagnetic energy from photons. Light reactions over here, dark reactions, or the Calvin cycle, over here. From there, I started talking with you about how cells talk with each other. It's a whole slew of different ways. I've listed them on that outline for you. Bacteria, it's done through a process, largely done through a process called quorum sensing. For our cells, there are all sorts of messengers that can come into play. There are phosphorylation cascades. There's other messengers within and between cells, like cyclic AMP. And let's leave it at that. Then, more recent history. I talked with you about how eukaryotic cells divide. The trick there is replicating the nucleus. The rest happens pretty much just as a matter of course, but going from one eukaryotic nucleus to a pair of eukaryotic nuclei is no mean feat. It's accomplished by a process that altogether is called mitosis. You can break mitosis up into individual stages. It starts with prophase. There's a point in time at which you achieve what's called metaphase. Then you go to anaphase and telophase, and you pretty much wrap the whole thing up. So I don't have anything here about that particular lecture, but here for meiosis, I've missed a lecture outline. But here we're talking about something I just started talking with you about last week. Meiosis is different from mitosis in that whereas mitosis makes two identical nuclei, meiosis takes a nucleus, has it undergo one round of division, of replication, very similar to but a little different from mitosis, and then without another round of DNA replication, ends up making a total of 4 haploid gametes, eggs or sperm. In the first half of meiosis, in meiosis 1, prophase 1 is the time when recombination occurs between chromosomes. That happens at synaptonemal complexes. The further apart two genes are, the greater the chance that a recombination event will take place between them. If two genes are on different chromosomes altogether, there will be a 50% recombination. They will sort independently. But if there's less than a 50% recombination between the two genes, that telegraphs to you that these are genes that are in some way physically tethered together. In other words, they're part of the same chromosome. There are covalent bonds that link them together. And I think that brings us, here's the the pictures of those things. And then that brings us to what I've just been talking with you about on Thursday and today. I don't feel I need to summarize that since it's just still hanging in the air. So let's leave it at that. I'll see you guys on Thursday, and then we'll be back to work next Tuesday.
I have a question. Okay, so with genetics, if I wanted to, or if someone wanted to eliminate a trait from society as a person, how is that even possible? To like say for earlobes or something, if someone wanted to have people with only attached earlobes, every time someone was born